Blood. Part 2. Circulatory system. Blood clotting. Blood loss from a torn or ruptured blood vessel is prevented when a blood clot forms to repair the damaged vessel. Fibrinogen, along with two other proteins, wrap around the damaged area and seal the cut. The threads form a mesh of threads that trap platelets and red blood cells. As water evaporates from the clot, it hardens into a scab. Blood clotting. A thrombus is a blood clot which forms within a blood vessel. If it breaks free and is carried in the circulatory system, it is known as an embolus. A thrombus is dangerous because it blocks the flow of blood to the body cells. An embolus can be very dangerous as it may clog an artery leading to a vital organ. If a coronary artery becomes clogged, coronary being an artery around the heart, it may cause a heart attack. A clogged artery in the brain may cause a stroke. Hemophilia is a hereditary disease in which one of the clotting factors is missing from the blood. This is actually an X-linked recessive trait, and that a carrier is female. Persons with this disease receive blood transfusions to counteract the effects of this condition. Blood groups. In the 1600s, blood transfusions were attempted. Some were successful, and others were not. The wrong blood types had been mixed, but it took another 300 years before they were recognized. In the 1900s, different blood types were discovered. Special markers, called geoproteins, acting as antigens, were found on the membrane of some red blood cells. In connection with the antigens, certain antibodies were found in the plasma or serum. The antigens were called A and B, and they gave rise to our four basic blood types. When blood antigens and antibodies mix, the blood clumps. This is known as agglutination, resulting in serious, possible fatal effects. For example, a person with blood type A should not be given B. The B antibody is already in A-type blood and will react with the B antigen of the incoming blood, and it will cause it to clot or form clumps. This is a serious situation that is easily avoided. Persons with AB blood are known as universal acceptors. They have no antibodies in their serum and have both antigens. They can accept any type of blood. However, they can only donate to persons with blood type AB. Persons with O blood are universal donors, as their blood contains no antigens. However, they can only accept blood from other individuals with type O blood. The following shows blood types, donors, recipients. The headings are blood type, antigen on RBC, antibody in plasma, who they can donate to, and who they can receive from. So we'll start with O. So O has no antigens, and it has A and B antibodies in its plasma. It can donate to A, B, AB, and O. It can only receive, though, from O. A blood. It has antigen A. It has antibody B. It can donate to A and AB, and it can receive from A and O. B blood. It can receive from B, antigens from B. It has antibodies in the plasma for A. It can donate to B and AB, and it can receive from B and O. Then we have AB, and it has antigens on A and B. However, it has no antibodies in plasma, and it can donate to A and B, but it can receive from A, B, AB, and O. The Rh factor. So we have 4 basic blood types, but we also have the Rh factor. So in addition to the ABO blood types, the Rh antigen was discovered in the 1940s. Individuals who have this antigen are called Rh positive. So there, who has this antigen? 85% of all Canadians are Rh positive. Individuals who do not have this antigen are considered Rh negative. And there are some problems that can arise with the Rh factor. And most of these occur with expecting mothers. So this antigen can present problems to expectant mothers and babies if the mother is Rh negative and the baby is Rh positive. Now remember, 85% of all Canadians are Rh positive. Nutrients and oxygen diffuse from the mother's blood into the baby, and the waste diffuse from the baby's blood back to the mother. This presents no problem because blood flows do not mix until birth. At that time, the mother's system recognizes the Rh antigen as a foreign particle and starts to produce antibodies. The baby is born before any problems can occur. So this is baby 1. If the same woman has a second pregnancy, there can be a problem because the mother's body still contains antibodies to fight against the Rh factor. So if a second baby is Rh positive, so baby number 2, the baby may not receive enough oxygen at birth, and the term blue baby has been coined to describe this condition. Treatments involve transfusions of Rh negative blood for the baby, or the mother may be given a drug that inhibits the formation of antibodies against the Rh positive antigen. This drug must be injected 72 hours after the birth of an Rh positive baby. Then we have artificial blood. It's known as Fusosol. It's a non-toxic liquid containing fluorine developed in Japan. It carries oxygen and carbon dioxide. It requires no blood type matching, which is a good thing. It carries what we need, and it's non-toxic. It can be frozen and stored for long periods of time. All good things. There's also no expensive screening procedures required before use for transfusion. Another good thing. Will not carry HIV or hepatitis virus. Another good thing. Will not clot and will not provide immunity. This is where it gets problematic. Provides time until natural blood can be given or can be used for transfusion for people with anemia, which is a good thing. And that's it.